All right, for our next talk this morning, we have Dr. Nathan Furey. He's coming to us from the University of New Hampshire, where he's now a professor in biology. Nathan was a Vanier Scholar at the University of British Columbia with Dr. Scott Hinch, where he did his PhD on bull trout, sockeye salmon interactions. Nathan's been a part of Ocean Tracking Network and ideaOTN for many years, so we're thrilled to be able to rope him into giving a presentation for us here. He's going to talk about his experience both in freshwater and the marine environment, tips from the field, so lessons he's learned in deploying arrays, designing arrays, dealing with challenges with data, and everything to do with how to get data from the field onto your computer, ready for analysis. Thanks so much for coming, Nathan. Really looking forward to your talk.
Awesome. All right, thank you so much. Just give me a second here to try to clip this sucker in. Awesome. Okay, I guess I'll hold this. So thank you all for being here. This is an incredible turnout, and I know they had to turn a lot of folks away. So I just kind of want to thank Rob and Kim for organizing. I want to thank OTN and ideaOTN for bringing me here, bringing a couple of my students here. This is my first time back in Canada since I left a couple years ago, so this is kind of a bit of a not a homecoming, but maybe a professional homecoming. And for just, before we get into the details here, I kind of want to let you know how wonderful of an opportunity you all have here. Because by being here, you guys can be a part of this OTN community or this OTN family. I started as a PhD student in OTN, and it really made my career. It funded my PhD, it helped me transition to a postdoc, and I know for a fact that the productivity I had during my time with OTN is what helped me land a job, and now we're working with OTN on several of our projects. So take this opportunity to become part of this family. Meet as many people as you can, get connected with as many folks as you can, and try to turn this into a lasting relationship if you can, because it really is worth it. Okay? So as folks mentioned, I'm at the University of New Hampshire. I think I'm the one speaker from an American institution over the next couple days, so on behalf of my country, I'm sorry. And so when Kim and Rob approached me, I'll admit I was a little taken aback, because they're like, well, you're an expert in deploying telemetry arrays. And my immediate response was, well, no, I'm not. And so really my job here is as much as to, not an expert, but I have had experiences in varied environments, so trying to tell you what's worked, what hasn't worked. And for those of you who have already deployed your own field arrays, you might not learn much, but hopefully you'll learn at least something, or at least it can kind of solidify your thinking and some of your own approaches or validate them. And if it is, mostly a review for you, I will poke fun at myself throughout this talk, so hopefully at least you'll find that entertaining. And so just kind of realize that my experiences are indeed very limited. I have worked on, the animals that I've used telemetry on include spiny dogfish, winter flounder, southern flounder, striped bass, bull trout, sockeye salmon, steelhead, and now my lab is involved on 2 different species of crabs, lobster, as well as Arctic char, and 2 different sculpin species. And yet the oceans and our marine environments and aquatic environments are so vast and large that that's still just a tiny drop in the bucket of everything that you could tag in every environment that you could track them. So I think kind of the ordering of the talks was really nice because the first talk by Cass et al., they were kind of focused on those big, large-scale marine arrays. And that is not where I've traditionally worked, or when I'm getting data from those, they've just been kind of handed to me, which is wonderful, and another reason why you should be a part of the OTN. But I tend to work in smaller systems in terms of where I'm actually deploying these arrays. So this is Christmas Bay in Galveston Bay, Texas. It's a barrier island. These are very shallow estuaries. We're talking maybe 1.5 meters or 2 meters deep at their deepest. Very soft bottoms, and we'll talk about that later on. These are some of the estuaries and salt marshes that we have in kind of this region, the Atlantic seaboard, rockier, greater tidal variation, etc. And then during a lot of my PhD, I worked at Chilco Lake and Chilco River and some of the rivers downstream of there. So that's kind of where my perspective is coming from. But hopefully we'll be able to kind of compare and contrast among some of these systems so that you can get an idea of how you might be able to apply these principles to your own study system. Okay? And so kind of my goals for this talk are to try to break it up into kind of two sections that we're going to focus on your study design, because all of the decisions that you're going to make is be driven upon what you want to accomplish in your research. And then we'll kind of go on, okay, once we've decided what's ideal for my research questions, how do we then implement that in the field? And again, this will be at a pretty coarse level. And so if we just think about a telemetry and what data we're trying to get from it, and then if we just got the ideal, as ecologists and biologists, what data would we have? Well, those data would be knowing where an animal is at all times and all locations, and we know what it's dreaming about and what its biggest desires are, and everything about the environment that it's in, right? Because we always want to know as much as we can. However, we only get a very simplified version of this. We're only getting locations at certain times that are often biased to what we're doing. We may know some things about the environment, but not as much as we'd like to. And so this is where study design comes into play. How do we maximize the resources that we have to answer the questions that we want to answer, while also minimizing the issues? And so before you start a telemetry study, I can't iterate this enough or reinforce this enough, you need to know what you want to know. What are the questions that you're going to try to ask and try to answer? And then what data do you need to answer those questions? And there's no shortage of the types of questions that we can ask with telemetry. It used to be a few decades ago, what we did with telemetry is you tagged a bunch of animals and then you just said, well, animal 1 went here, and then it went there, and then it went there, and then it went there, and then it came back. We have advanced the technology, we have advanced our thinking, we have advanced our analytical tools that we can answer a whole lot of different questions. And so I'm not going to obviously go through all of these, but there's some really good reviews out there that kind of discuss all of the things that are being done with telemetry, both from basic science, so biology and ecology, but then also helping us to conserve and manage populations and to solve applied science questions. Just another example of the varieties of data that we can get from telemetry, and this was alluded to in the first talk as well. We get data in different dimensions and at different spatial and temporal resolutions. We can pair that with environmental information. We can pair that with physiological information on the animals. So we have no shortage of questions, and so that can be a little bit daunting if you come into, let's say, a master's or a PhD program and you're just told, well, I'm going to tag a bunch of species X and see what it does. You need to think a little bit more about what exactly do you want to figure out before you go out into the field. Okay? So what is your question? And I want you to think broadly. I want you to think in terms of biology, ecology, or you might have a specific applied problem that an agency or a funder wants you to look at. But I don't want to minimize those natural history observations. It's still important just to know in a lot of systems that we haven't done much work on these species with. It's just important to know where these animals are going, but hopefully then you can start to build and broaden. So those of you who are fortunate enough to have multiple field seasons on the same project, don't be content necessarily with just redoing the same thing that you did in year 1. Try to go back, look at the data that you've collected. Is there anything that we could add to it to better answer the question that we're asking? Okay, so kind of moving on. Once you've decided what your question is, you're going to start thinking about the receivers that you're going to use and kind of the geometry of that array. And whenever I'm kind of discussing with students array geometry, I always start with this really important here by Michelle Heipel. Because I think some of the figures and some of the concepts in that are really important. My guess is most of you in this room have read this, but if not, go take a look and I'll be pulling some figures from that here. And so this is the idea that if you have an environment, you can put receivers in that environment in all sorts of different geometries. But realize that geometry is going to dictate what types of data you get back, as well as what types of questions that you're going to be able to answer well. So kind of the two classical approaches that we can see here is a grid. It's just kind of whether it's a random grid or a stratified grid. You kind of just try to cover as much area of the environment as you can, and that way hopefully you're maximizing your coverage, which in theory, if the animals are moving at random, we know that they don't, but you'd maximize your chances of detecting these animals through time. And then we also have what we call gates or lines, where, you know what, an environment can be really, really big. We're not going to be able to track them through time. So we just need to know if they cross this point or they get to this point. And this can be really useful for animals that we presume are moving in a linear fashion. So let's say migrants, because they hopefully are going to have to pass that line at some point, and then you'll be able to say for certain whether you got them or not. While a grid might be better for certain just descriptors of habitat use, habitat associations. Yes. One of my pet peeves that I see in the literature and that I see in proposals, however, is that people assume that if you have this line and you get a detection there, so let's say you have two lines, and you get a detection here by an animal, and then hours, weeks, months, however big this distance is, you get a detection there. And the assumption by the researcher is that, yep, left the area, right? Started here, and then went up to the north end, and then it's gone, right? That can be a really dangerous assumption. Realize that there's a lot of different potential pathways that an animal can take and still give you the same data in terms of detections. Right? So if these are all receivers here, and these are all different paths of animals, and if we assume that the little points fall in the line, that you're getting a detection. And so certainly, sometimes when an animal is moving, it can be linear, and you'll get a few detections at this array, and then yes, it did indeed go off. But maybe it went through and came right back. Or maybe it went up to your line before returning back. And so oftentimes we see these lines deployed on, let's say, management boundaries, and then we use that to then say, oh yeah, this animal passed from management boundary A to B. Are you sure about that? And at what spatial resolution do you need to define that at? This might be close enough to say, you know what, if it gets here and we're right on the management boundary, we can say that it's using both. But just be careful when you're interpreting your data. And so because of this, and this will be kind of a message that I'll repeat in this talk, redundancies in your design can be really valuable. And so that includes building double gates at really strategic locations. If you want to know for sure that they have passed a boundary, you might want to have a gate just before, or a line just before, and a line just after it. That way you can at least continue that linear movement a little bit further. This is also, I won't spend a lot of time on kind of mark recapture studies and survival studies of animal migrants. I just didn't think we'd have time, and maybe stepped on the toes of some of the analysis stuff. But realize if you are using mark recapture models to estimate survival of an animal along a route, and you're estimating detection efficiency within those models, you're not going to be able to estimate a detection efficiency at your final array. So if you build a double line at the back end, you'll at least be able to get the first part of that array. And I can talk more with folks about that who are doing survival modeling later on. Okay, so if we go back to our hypothetical figure here from Heipel et al., you know, I'm just kind of talking about the geometry here, but we have those dotted lines around them. And so probably most of you are familiar with the idea of detection radius or detection efficiency. And it's nice and really easy. I mean, it's just like one click in R or in ArcGIS to just build a buffer around each of those receivers and assume that there's a set distance at which you can detect an animal. However, this is really not sufficient. And so you need to be able to determine your, and I know they're two different things, but I tend to use detection radius and detection efficiency kind of simultaneously because they're kind of the same concept. But you need to estimate that whenever you can. And I know Jake is going to get into this a little bit tomorrow, so hopefully this is seen as a bit of an introduction to that. But this is the idea that when you're really, really close to the receiver, you can hear the tag quite often. And the further you get away from that receiver, the less often you'll hear it. And so defining kind of the shape of this curve, as well as just kind of knowing the general area, when do you have a really good chance of detecting this tag, is really important. And so this radius is largely dependent upon tag power, and kind of the decibel output of it, the the folks who build this would be able to talk about that in much more detail with you. But some of our estimates in really small tags, so something like a V4 or a V5 in, let's say, a somewhat noisy environment, you might only get 50 meters of detection radius, while some of your really large tags you can get a kilometer or more. So again, this is a decision that you're going to have to know what size tag are you putting into your animal, and that's going to affect the spacing of your array. And so even if you were to do a range test, and we'll talk about that a little bit, there's kind of some assumptions that we tend to use. And those assumptions include that the detection radius is constant among all of our receivers. So it's all right, well, I tested it in one part of my bay or one part of my river or one part of my lake, and that detection efficiency or that radius is going to be the same throughout the study system, as well as constant through time. All right, I tested it, even for all of my sites, I deployed it, now I'm good to go. There's also the assumption that certainly, as we just alluded to, that the power output of the tag is going to influence that detection efficiency or detection radius, but maybe not. There's probably not a difference between animals or maybe tagging method. Now, we're all a lot of really smart people in the room, and biologists and ecologists, so I think you can probably say that, you know, we use these assumptions a lot because it's just simple and it simplifies our analyses, but there's probably some things that just don't register with that in a realistic basis. So that we do need to try to assess these things on a study-by-study basis and by location basis, as well as through time. So things that are going to affect detection efficiency or detection radius, a lot of it's noise, right? If you're talking about, and I am talking about acoustic tags here, if you have a lot of environmental noise, whether it be natural or anthropogenic, that can interfere with your signal and attenuate the signal and reduce your detection efficiency. Realize that some environments just have a lot of biological noise as well. I don't work in coral reefs. I somehow end up in cold places. But there are certain environments that are just very loud and noisy. And this can also not only change from location to location, but through time. And so you need to at least be aware of these things. You also need to remember that tags and receivers need to maintain a line of sight between each other to get a detection. Now, this isn't always true, but this is kind of, in an ideal world, you have a tag and you have a receiver, and there's nothing physically blocking the line of sight between the two of them. And so this is something that we deal with a lot in riverine environments, is thinking about if you have a windy river, you might have a really good location to put a receiver, and the radius and the acoustic quality might be really good for maybe hundreds of meters, but because there's a sharp bend in it, you're really only going to be able to detect that animal for maybe 50 to 100 meters. And so you're going to have to choose your locations in that river appropriately, or at least recognize that that is going to be an issue. And we'll kind of talk about tag programming and touch on that later. Again, I don't work in coral reefs, but you know, one of the most basic characteristics of a reef that you can measure is the rugosity, you know, the three-dimensionality of it. There is a lot of structure to coral reefs, as you can see in this very, very fuzzy image that I stole online. But the idea here is that if a fish is in one part of the reef versus another, relative to your receiver, when you don't get a detection, that might not mean that the fish isn't within your radius. It just means it's blocked by that physical barrier there. And so you do have to be really careful about where you're placing your receivers and trying to think about how can I maximize my line of sight just in terms of total aerial coverage, but then also thinking about that animal's biology as well. And so all of these things interact to generate a lot of variability in detection efficiency. So this is from a paper that was looking at detection efficiency of acoustic receivers in a small river system. You can see the spatial scale here. We're only talking about like maybe 2 kilometers distance here. And kind of you can think of it as red is bad detection efficiency, green is good detection efficiency. So even just over the course of a kilometer, you can have some sites that pretty much don't work at all versus receivers that are getting all of the detections that are available to them. So you need to think about this as you're deploying. Okay, so if this wasn't complicated enough, at least this is all factors that operate over space. And you can go out and figure out where the barriers are. You can think about where your critters are going to be using the habitats and try to maintain line of sight. But then also realize that detection efficiency is going to vary through time as well. So even in a given location, its detection radius is going to change through time. Okay? There's a couple, and there's several well-published papers on this. I think Jake is going to touch on this as well. But this was just a study that was showing how the impact of wind. So I think this was in a lake system. And so simply the stronger the wind, the greater the wave action, the greater the turbulence in kind of the surface waters. I'm not a limnologist or oceanographer, so I probably butchered the physics and terminology there, but you're creating a louder environment that then makes it more difficult to hear detections. So here we just simply have the number of detected transmissions across wind speeds, and we get this negative relationship. And then these are just simply predicted values here. So essentially, if your detection radius is going to decrease as you have higher and higher wind speeds. Okay, so how do you deal with this? And again, Jake is going to touch on this quite a bit tomorrow. But you can do range testing. So this is actually as you're deploying or shortly after you've deployed, you can go out and try to assess what's your detection radius. This is really good for understanding those spatial factors. However, it's not as good for, let's say, those temporal factors because you're kind of testing it once. Okay, my geometry's set, now I leave it. You can have what are called reference or sentinel tags. So this is, I think, getting alluded to in the first talk. The idea that you can just simply put tags in strategic locations within your array. You know their locations. You know how far they are from your receivers. And then you can just simply see how well are you detecting those tags throughout your entire study. So if you start to see, ooh, this week or this day or maybe every night, we see that we're not getting our reference or sentinel tags as well, that might give you an indication that there's some other temporal factor there that's going to affect not only how you detect those receivers, but all of your tagged animals. And then again, we can use stats to help us out a little bit as well. So if you're doing something like survival modeling, you can actually use detections of, okay, I know how many animals made it to this line. I know how many animals I did not detect at this line, but were actually detected on later on in their migratory trajectory, and we can use that to actually estimate detection efficiency. However, these still don't properly, at least the ones that I'm aware of and know how to use, there's probably more complex ones available, don't necessarily incorporate that temporal difference that you get throughout the study. Okay. All right, so detection efficiency can change in space, detection efficiency can change in time. You also need to think about how many critters you're putting out into the system and how much you expect them to move, right? These tags rely on these pings going out and these pings getting recorded on a receiver. If you have a lot of things pinging at once, there can be confusion or collisions, and you're not going to get either or any of them. And so this is just from this paper here, the probability of collisions with the increasing number of transmitters within range of a receiver or an area. And so Vemco also has a calculator online that you can kind of use to, you can kind of say, okay, this is how many tags I want to release. This is, I think, the area. I haven't looked at that calculator in a long time. But anyways, it can try to help you think about the idea of how many tags should you be putting out at once. And like one of my messages in this talk as well is that there's no perfect study, right? That especially when you talk to Vemco, they can be, and this is not to say anything bad about Vemco, I love Vemco, but right, they want you to maximize the data you get per tag. Because why wouldn't they? That looks great for them. And so they try to, again, maximize the data you get per tag, but then you as a researcher are going to have to decide what's feasible. And so I want to give you an example of this. So this is kind of a lake-river entrance between, so this is the lake here, and then these animals are going to be moving between this lake and the river that are up there. Each black dot is a bull trout. that we've tagged. This was a VPS study that we did. And this video is over a 24-hour period of these bull trout. And it starts at noon. And what you can see is that there's a lot of different dots. And that's because we're getting good position estimates of these animals throughout the day. We do not have too many animals in the system at this time. However, we weren't just tagging these bull trout, we were also tagging sockeye salmon smolts that were migrating through the system between 30 and 40 a night. And so those fish would be moving through right about now. And you can see now we're only getting like maybe 6 or 8 of those dots when before we were getting 12 or 15. And that's because we've just put in another 30 to 40 animals in the system with them. And you know, if we had told Vemco we were going to do that, they'd be like, no, don't do that. But we're only up there for a migration that might only last for 3 or 4 weeks. We never know each night how many fish we're going to get. So we had to make the decision, what's the maximum number of fish that we can put out and make sure that we're going to get all of our tags out for the season while also minimizing the tag collisions. So we tried to minimize it to on the order of 20 to 30 tags a night so that we could get as good of data as we could. But you can see that we had a loss in the quality there. We still think that we have enough data to say something really meaningful ecologically, and there is kind of a cool story to talk about there, but I won't because that would be too tangential. But anyways, you need to be cognizant of, you know, a greater sample size might not always be better, especially if your animals are using a confined space. Okay. How long are you going to track your animals for? You figured out maybe the best geometry of your array, but how long do you need to track them for? You know, is this a short seasonal migration that's only a few weeks? Or do you only have enough receivers to cover a small area, so you just assume they're not going to stick around very long? Or is this going to be a year or a multi-year study? Or is it going to be a hybrid? Or are there certain areas that you're going to cover with an array for maybe a month or two, but then there's other sites that you're covering for a year or more? The duration of the study is going to affect a lot of your decisions. It's going to affect how you deploy your receiver moorings. It's going to determine what tags you use and the programming of those tags. So I'm actually going to start with the bottom one, be unintuitive. You really need to think about, so the decisions with how often a transmission should be sent out. The longer that duration between successive transmissions, the more battery life you're going to get. And so oftentimes people want to do that. They want to push that battery life longer. But you need to think about the geometry of your array and the environment that you're in. And think about how long will a tag or your animal be within a detectable radius. Another way to think about this is how quickly is that tag going to move by your site? And then thinking a little bit further, how many potential detections are you going to get per animal that you actually think you have a good chance of getting detection? Okay. So this is going to be affected by the environment. We have to think about this a lot when we're studying migratory salmon. Right? So we are tracking migratory salmon over 1,000 kilometers on the west coast, and when they're in these small river systems, they can still travel 30 plus kilometers a night, and then when they're in a big river system, they might travel 200 kilometers over 24 hours or more. And when you start to do the math and you think about how small and windy these rivers are, you quickly realize that if you have a tag programming of over a minute, there's a really good chance that that animal or that tag literally won't ping during the entire time that it's through your detection radius. And realize this is where you can also get creative with the Vemco folks or whoever you're getting your tags from, and that you can set your programming at multiple stages. So for example, for our migratory salmon that are going through really windy rivers, and they're doing it pretty quickly, we have a really rapid transmission, maybe every 30 seconds or so. And again, we have to balance that with the whole issue of tag collisions. So again, there's no perfect study. You're just trying to minimize the weaknesses here. But then, after a week or two, they're in the marine environment, and they slow right down. So then we have, after 10 or 14 days, we slow those tags down such that we can maximize their battery life as they go up the coast. So these are all important decisions that you have to make. And then it's also, obviously we've talked about tag power is going to influence that radius of the receiver, and so that might give you a longer duration of time that you expect that animal to be within the range of the receiver to be detected. And then obviously animal behavior. If you've got a slow-moving animal, I've seen some studies that are doing things on like whelks or other shelled mollusks that have a really small home range. You might be able to really increase the duration of the study by jacking up that time between transmissions, because you only expect it to move a few meters a day type of thing. But what you need to ask yourself is, what's going to happen if you miss a detection? And at each location or each part of your array, if you miss that detection there, are you able to answer the question that you want? We're never going to get all of the detections, but you need to prioritize locations and think about this. And so one of my tips is that if you have a location that's really important, use more receivers than you need. And I'm putting need in quotations there, right? And so some people, again, going back to that hypo et al. Figure, they just want to spread all the receivers out as much as possible to cover as much ground as they can. But depending upon your question, it might be better to just focus on a few key areas to maximize your chances of getting a detection there. So this is a map of the array that we use to track those juvenile sockeye salmon as they left Chilco Lake. They then go through the Chilco River, the Chilcotin River, and then the Fraser River, and then they travel through the Strait of Georgia. Now, obviously, the NSOG line and the Queen Charlotte Strait line, those are all maintained by OTN, and those are kind of our traditional marine arrays that, again, I don't touch, thank goodness. But then you see all these dots here, Henry's Bridge, Siwash Bridge, Farwell Canyon, and then these locations down in the lower Fraser River, and they look like single receivers because there's a point there. Well, no, there's multiple receivers there, even though, in theory, a single receiver at Henry's Bridge should be able to do the trick. It's really narrow, maybe only 100 meters wide. It's a little bit deeper. We think the acoustic properties there are quite good. We could get away with one receiver, but if we don't get that location, we don't get anything from the Chilco River. So we make sure that we put at least a couple receivers there. And we do that at each of those locations, because if we lose any of those locations, we're not able to talk about an entire river in many instances. And that's really important to us because we're trying to figure out which landscapes survival was higher or lower in. And we'll talk about an example of this, that sometimes the environment doesn't agree with what you need as a researcher, unfortunately. Okay, and so again, no study is perfect, no tag programming is perfect, no array is perfect, and you end up sometimes having to make really difficult decisions. And so in the last couple years of this study, we actually started using V4s to tag the smallest of these sockeye salmon smolts. Sorry, I'm showing you a picture of dead smolts. I was lazy. And so what we have are the red dots are the V4s. And my goodness, it took maybe 20 to 30 hours of meetings and tens of hours more to figure out the tag programming on these, because they don't last nearly as long as the V7s that we were using in the H2 fish. So we needed a rapid transmission rate here in the upper rivers, because we know that they're moving pretty quickly. And then we actually estimated a short time that we expected them to be in the lower river in this lower part of the Strait of Georgia. We actually had them turn off for a while and then try to turn them back on so we could hopefully get those fish in this region here. And so you have some years that are just simply trial years. You make really educated guesses, you work with Vemco closely, but in the end, these tags have limits to them, and we have limits to our own brain capacity as well. And so you can take that information, learn from it, and try to move forward. But you need to understand that those decisions have consequences. Henry?
Are those tracks imputed?
Yes. Yep. Yeah. So we're only getting detections at the arrays, and then simply the migrations between are imputed. Okay. All right. So now you've put a ton of time and effort into trying to understand what you want to do. You have a good sense of the environment and kind of maybe the array geometry that you should use to specifically get the types of data that you need. But before you even go into the field, you need to ask yourself, how are you going to analyze your data? And so this is just a one-slide pitch maybe for the analysis, but I thought for the analysis session that you're going to have, but this is really important because oftentimes students, you have really great creative ideas, you get a bunch of data to come back and you're gung-ho about answering a few specific questions, and then you realize, oh man, these data can't answer that question. And so a way to force yourself to think about, can my data answer my question, is visualize your figures. This is something I tell all of my students to do, is that if you have a hypothesis that you want to test, you should be able to draw me a hypothetical figure that demonstrates it. You know, are you going to test your hypothesis? Is it going to be a scatter plot that you can then draw a regression or a generalized additive model line through it? Or is it a box plot that we can see, you know, is there a factor on the bottom and we can see where the different distributions of data lie? What is it going to be, a model selection approach, a multiple regression model? Those types of things. Because if you can visualize that, and you can visualize what goes on this axis and this axis, you can then say, am I getting those data from my study? But instead, if you just say, well, I want to answer this big, broad question about biology or ecology, I'm going to collect a bunch of data and then see what I can do with it afterwards, you're often going to be disappointed. Okay, so that's my one-slide pitch on think about statistics. Okay, so now that you know what you want to do, you know what data you need, you're confident that you can do the analyses that you want to do, now you need to go out and do the thing. So can you actually do it? You know, you have ideal locations in your head. Can you actually get to those spots? So this action shot of myself during my PhD days, this is at Farwell Canyon along the Chilcotin River in British Columbia. And to get to this point, we have to pseudo-rappel down a hill and then put yourself into the water where the water is moving very quickly and put a receiver down. That was kind of certainly my personal limits. For someone who does not enjoy exciting things, this was frankly terrifying to me. It's one of these things that, you know, leading these field expeditions, you know, everyone else could go pseudo-rappel in maybe about 30 seconds. For me, it was about a 5-minute period. I'm terribly afraid of heights. And so we usually would just take a 10 or 15-minute break once I got down to the bottom. But at least we look good while we're doing it. So you need to think about, how are you going to access these field sites? Who do you need, not just, you know, the physical difficulties, but who owns the properties there? Who do you need to be talking to? Do you need to reach out to, and you know, if you're working with First Nations or working in First Nations territory, you need to be talking with them from the get-go, even before you get to this point, to make sure that you have access and that they're on board with everything. But then also, what do you have at your disposal? You know, what is your budget for moorings? What is your budget for gear? How many people are going out there with you? This would not happen if it were up to me by myself. It would probably not happen if there was only 2 people. I would convince them that this was not a bad idea, or that this was a bad idea. And so this gets to the idea that sometimes these most strategic locations, in terms of what we want to answer as a biologist, don't match what's going to be effective for telemetry. So I'm going to go back to stinking Farwell Canyon, British Columbia. Right? This is where the Chilcotin River is just about to meet the Fraser River. So if our idea is to try to compartmentalize survival of these migrants among river systems, this is a really important one. Because if we don't get that, we don't get them again for hundreds of kilometers later on until they're bottom of the Fraser River. So we have to go down the hill. But unfortunately, we have to go down a hill because this is indeed a canyon. This is Farwell Canyon. So Adam might be able to correct me, but I think our receiver site is around that corner there. And so, well, guess what? Canyons aren't necessarily conducive to acoustic telemetry. They are loud. The water is quickly moving. And so we had multiple years that we put receivers out here and we pull them back up and we get fewer or no detections. And that's not because all the animals died. We got lots of detections further on downstream. So this site wasn't very good. And so this year, I'm wearing the dry suit. We had this great idea that we were just going to scope out the area and we were going to find a calm pocket of water. There had to be one, right? And so I'm also terrified of quickly moving water. I know that's not intuitive for a fish biologist. But so I was having to get in the water and actually work myself upstream in this canyon to actually find a deep pool to put the receiver in. And we'll talk about that later on. We'll tell you how it goes. But the message here is that no matter how important they are biologically or ecologically, there are some locations that just simply aren't good for this. Right? Telemetry is a tool. And not every tool works in every situation. So you need to think about how well is it going to work? What are the currents like? What is the anthropogenic noise going to be like? Is it going to be just super windy and you're in a shallow environment all the time? Also, you have to think about your taxa of study. Would there be a different type of telemetry that would be more appropriate for this animal? And so these are just some of the types of things that you need to consider, or maybe types of environments that acoustic telemetry doesn't necessarily work as well. I'm putting an asterisk on large pelagic systems because obviously you can use acoustic telemetry in large pelagic systems. Is just realize that you're dealing with a very, very big habitat and these tags and receivers only have a very small range that they can be seen. Sorry, I'm messing with the mic. So this can all seem really, really daunting. And I don't want the lesson or the take-home here to be that acoustic telemetry is really hard and I shouldn't do it. You can sometimes use all this information to your advantage. Sometimes you can answer a question with very little. And I want to show you an example of that. Okay, so we're going back to British Columbia. We have this same lake and river transition. And so I showed you the fine-scale movements of those bull trout that took a VPS array of something like 45 receivers, and we tagged 25 bull trout, and we tagged, like 400 sockeye salmon smolts that year. So a very complex array. However, we had a new question that was quite simple that we wanted to ask. Because we noticed that these bull trout were hanging out with these salmon, and in fact they were eating a whole lot of them. And it's all they ate during this time that these salmon smolts were migrating. So we had a really simple question of, do bull trout hang out in this area of the lake outlet year-round? And if not, do they come back the next year? So we had a very specific question, focused on a very specific geographic region. Just simply, do they leave and do they come back? So these two dots here, those are two receivers. Two receivers that we used for a whole study. And we picked two locations that we knew from a lot of testing that they had really good detection efficiency. And we simply were able to track that, yes, pretty much all the bull trout left either towards the river or towards the lake, and that we saw about 40% of them return the following year to exploit these salmon smolts. And we got a paper out of it. Two receivers. Okay, so now getting into more of the actual field implementations here. So your moorings. Again, we just talked about all these deep pelagic moorings with acoustic releases and 200-pound anchors and such. You need to determine what you can use for your study. And I am very, very fortunate. I have worked in some very easy places to work in. And I've also worked on migratory fish that are only hanging out for a month at a time. So sometimes you can get away with really, really simple moorings. This is a sandbag. The receiver zap strap to it. Repeat about 40 times. And that is your network type of thing. It's cheap, it's effective, we can easily get everything back. Here is another really high-tech version of a mooring that we use, a cinder block wrapped with electrical tape and duct tape around it. But again, this is deployed in a relatively calm environment for a short period of time. And it works. So this isn't to say just go really cheap on everything, but if you can get into a system, test how it's doing, don't make it more complicated and more expensive than you need it to be. Another example of keeping it simple. I mentioned during my master's I was working at Texas A&M, and I was doing some VPS studies of juvenile southern flounder. These are these really shallow estuaries associated with barrier islands, and the substrate is super soft. It's all essentially a muddy sand. So what we found was highly effective for our telemetry studies was buying 10-foot pieces of PVC pole, drilling some holes in them, zap strapping the receivers to it, and then pounding about 6 feet of that PVC pole into that sediment. And boy, that thing was not moving. We did this for even longer term studies, upwards of a year, and those receivers did not move. Because that sediment's so soft, it just fills up the PVC and you're good to go. However, sometimes you need to do more complex things, and again, if you're working in kind of deeper environments, there's other folks here that are going to be able to help you with that and get examples of that. This is kind of we went with a slightly different version than this, but this is kind of a rebar fabrication that we've used to try to maintain a low profile in rivers. We've had mixed success with it. You can talk to Adam about that later on. Some sites it's worked, some other sites it hasn't worked. We're actually using, in kind of the Gulf of Maine right now, in some of our work out of the University of New Hampshire, we're actually using modified lobster traps that we've kind of modified to have these receivers. There is a bit more variation in the bathymetry there, so we actually wanted to prop the receivers up a little bit to maintain line of sight. We'll talk about when this works and when this doesn't work. Realize that you need to think about not only just keeping the receiver there, but how the environment might affect your ability to do that. This is from actually a nice technical report that has guidelines for acoustic telemetry. I found it pretty informative. You guys can look that up as well. But if you are an area that you need to have a surface buoy because you need to be able to locate it, realize that buoy is going to get pushed and pulled around by currents, by wind, people can mess with it, etc. So if you have physical forces acting upon it, you might not want to have your receiver directly attached to that line, particularly if you're doing something like a VPS and you need to have really precise locations. So you might have a daisy chain or a chained type approach to mooring your receivers. But again, your need to do this is going to be dependent upon the environment. We've done this with a lot of our lake receivers that we know get a lot of wind action, but then in other locations that we don't think there's strong tidal or wind currents, we can just simply use a mooring straight to a buoy and a receiver. This has already been mentioned, but you need to think about the environment and biofouling. I'll just admit I have used the biofouling paint before. I know it is not good for the environment, but it has worked for us in the past, so you just need to make that decision on a case-by-case basis. Realize if you're working between freshwater and saltwater, I think most people here, this is the Ocean Tracking Network, most people are marine folks anyways, but realize if you're transitioning from a mooring in saltwater versus marine, that rebar type structure probably is not going to work in an estuary or marine environment. We've already mentioned currents and winds. Also think about the depth variability. I talk about this in two regards. The first is the tidal cycle. So down in Texas, the tidal cycle isn't very big in terms of the magnitude. It's only about a foot and a half between low tide and high tide. But if you're talking about working in an estuary that's average is only 2 or 3 feet, that 1 foot is going to make a big difference. And so we actually had certain habitats that we kind of just had to say, you know what, we're not going to be able to get detections during this time. So not getting a detection in these certain areas at certain times of the day didn't mean that the animal wasn't there. And so that's something that you can track and again try to look at whether it's your reference tags or etc., sentinel tags to try to assess that. However, realize there's also seasonal flows. And this is more of a river type concept. But we have certain locations for our bull trout work that we would really like to put a receiver because they're potential spawning locations for these bull trout and we want to get a sense of what they're doing year in and year out. However, when the water levels are really low, these receivers would be out of the water and it would be freezing temperatures. So that's not really a risk that we're willing to take. So you need to think about what are the conditions that receiver is going to experience from the time that you drop it off to the time that you pick it up. So that kind of gets to the freeze-thaw. Also the idea of flashy rivers and sedimentation. So again, in the bull trout project, and so we had that picture of brave Nathan going down the side of the mountain to get data. That's how much I love data. And got in the dry suit and wiggled my way around, and we actually found a deep hole that the water didn't seem to be moving very quickly in. It was like 20, 30 feet deep at the bottom of this canyon. And, you know, again, I'm not pretending to be smart up here. I just have a lot of experience. And so we're like, this is great. It's deep, it's calm. Let's put a receiver down here. Well, I'm sure some of you are already thinking this in your head. Well, you're at the bottom of a canyon where the water's moving really quick, and then all of a sudden it stops. Everything that comes down that river, down that canyon, is also going to stop, and it's going to fall. And so we were able to go back and find the buoy, and you pull, and you pull, and you can just tell that there may be literal tons of material on top of this. We have other locations for our bull trout project where rivers meet, and you have convergences. And again, from a biological standpoint, this could be really helpful because you can see whether this animal goes from one river system to another. Well, this is again where waters converge, and the physics can slow the water down. This is also in British Columbia. These rivers are really flashy, so you have times of low water, and then you have big pulses with the snow melt and the spring rains, and it brings a lot of sediment down with it, and that sediment's got to stop somewhere. And so, Adam, I know you've had at least one receiver that you were able to find the location of it, but weren't able to, well, I think you were actually able to get it out of a meter or two of sediment.
About an hour.
Yeah. So that's dedication to data, you know, dig for an hour through straight sediment. Something that we haven't mentioned yet is the idea of human intervention. This is something that you need to know. You need to know the community that you're working in and the different stakeholders that are in the region. How likely is it that the presence of your gear might upset somebody? And are you reaching out to those folks? Is there a way to try to communicate with them that this is for the greater good that we're doing this research? Oftentimes saying that you're not with the government can help a whole lot. But if that's still a big risk, then do you make decisions such that your gear is actually a bit more conspicuous and a little bit harder to see? And I think I touch on that later on. Okay, so you've got your mooring, you've got it robust enough for the location that you want. Just remember that even if you're not doing a VPS and you're doing kind of presence-absence studies, remember that you need to maintain that line of sight, and just a little bit of spatial variation from one location to another can actually change your array quite a bit. So be really aware, again, of those spatial variations in line of sight. Oh, here, we're back to security of your receivers. So this is the Chilco River in British Columbia, and kind of the person who's taking this image is actually standing on a bridge. And that bridge is really popular type of thing. And so we had some reports of folks at least looking at the gear, trying to figure out what the heck is in this river, and pulling it out. At least they came and talked to us about it, so that was great. But we kind of decided, you know, to make sure that folks don't get too curious, we actually weren't going to mark our receivers. And again, you can only do this in certain locations because it's shallow, it's slow moving, we can wade, we have really good GPS points of it all, but we kind of had some landmarks, like a big rock, and then there's like a fallen tree, I think back up here, and then you just walk out into the water, dump the gear. We have dry suits and waders so that we can do that type of thing. So you just need to think about that, because we've lost several receivers due to folks just pulling them. Or people are just curious, they see a buoy and like, oh, what the heck's this? I've never seen this before. They pull it up and they're like, well, can't do anything with this, and then they just dump it on shore. Which isn't the worst case scenario, because at least you get it back, but you just lost all the opportunities for data. I mean, we just recently had a study where a lobster fisher literally told us, I pulled it up, saw it was from your university, we don't like researchers, so I just drove it to a random area and dumped it off the side of the boat. We somehow found that. I have no idea how. I really thank some of our logistics folks for that at UNH. Okay, so another opportunity to laugh at me. Particularly in small environments, sometimes you can get lazy, right? And you can just be like, well, this water's shallow. I have enough rope on the end of this mooring. I can dump it over the line, dump it over the boat. We really thought we had enough rope on this mooring. But as you can see, you can't see the buoy. It's actually it'd be under the boat. And as the not so fearless leader, it was my job to get it back. So I was literally kind of not free diving because it's awkward in a dry suit, but trying to get myself down as low as possible until I could actually grab that rope and then try to actually heft. They were then hefting on me to get that mooring back. So measure the darn depth. Okay, and measure it twice. And this is, I know it looks beautiful and blue, but that water is probably about 4 or 5 degrees. Okay, so now you've deployed your gear, you measured the depth, everything is out nicely, but let's kind of try to bring it back to this idea of how well is this array working. So this gets back to the idea of range testing. There's a few different ways to do this. Experimental tests. So simply you have a receiver, you can measure out set distances from it, you can put tags there, you can leave them out for a while, and you can just see how many detections are you getting. If you have a VPS, that's a little bit more dynamic, right? Because it's not just how is each receiver doing, it's how then does the coordination of multiple detections result into an estimated position. You can do what's called a drag test. So set up your VPS and then actually take a tag and actually run it through your array while you're also tracking a GPS track. So you have your handheld or the boat's GPS, and so you know the location of that tag as you drag it slowly through your array, and then you upload those data to Vemco, and they're going to give you estimated positions for those tags, and you can compare that to your GPS track type of thing. And then we have the reference or sentinel tags. This gets at more of the temporal variability. These first two can get at kind of that spatial variability in detection efficiency. But then having tags out in the environment through the entire study's duration allows you to get a sense of kind of the temporal variation in the quality of your detection efficiency. But I'm going to give a bit of a disclaimer about reference and sentinel tags here. And this actually gets into the idea that how you tag your animal is going to affect your study. And so I think a lot of us are fish biologists, so I'm biased towards that. But most often we're doing internal tagging, right? That tag is going inside the animal's body to stay for the duration of the study. We can also externally tag. This is one of our Jonah crabs that we've given a little fancy backpack to. And that you can externally tag fish as well. We've done this a lot with flatfish, especially smaller flatfish. They don't have that internal body cavity to really accommodate a tag, so we end up giving them a little backpack as well. And there's a lot of reasons, and we'll talk about why most people do internal implantation. Most cases, I suggest that you do use internal implantation. However, you need to realize that that's actually going to affect some aspects of your study. So this paper is kind of a brainchild of colleague Mike Dance, who's now an assistant professor at Louisiana State University, as well as myself. When we were very, very green graduate students, we had never done our own telemetry study before. And Mike always has kind of crazy, harebrained ideas. And he just said, he's like, well, doesn't putting this tag in an animal affect how far that signal can go? And we're doing range tests just with the tag outside the animal. Is that really the same thing? And I'm like, I don't know. Let's ask some people. And we asked people, and kind of the answer that we got was, well, the fish's body is very similar in chemical composition to water, so the sound flows through it. But then in the same conversation, they would tell us that things like seagrass would affect the detection efficiency. And those two things did not really compute or add up in our heads. And Mike is also a very stubborn individual, which is a good thing. I probably would have been like, screw it, let's go with what they said. But Mike's like, no, we're going to do an experiment here. And so I'm only going to really focus on kind of experiment 1 here, but simply taking a red drum, which is a popular wreck fish down the Gulf of Mexico, and internally and externally tagging it, putting it in a cage with known locations, and then we're going to set up receivers. So we're not going to move that fish around as you would maybe or that tag around as you would in a traditional range test, but instead we're just going to use a bunch of receivers at varying distances in this shallow estuarine seascape, and then see what happens with detection efficiency. And this is what we get. The internal tagging is the gray. The external tagging is the black. Now granted, we only did this in a very specific environment with only one type of fish. So whether this translates to all these other studies, I'm not sure. But certainly we had very strong evidence, and we saw this in several trials and several ways of doing things. We got the same result. And so that idea that if you're tagging internally, but then you're using an external tag as a reference or sentinel tag, it's still going to give you an idea of the relative quality of the performance, but is it going to give you an absolute estimate? I think that we need to think about this a little bit more. Robert?
Does that influence how you conducted range tests?
Yes, sorry. So the question from Robert was, has this result, this knowledge, changed how we do range testing moving forward? In some cases, yes. But realize that it's difficult. We do range testing in the ways that we do because it's logistically feasible. And so when you're telling, let's say, IACUC or animal care that, well, we need to take this animal and actually hold it in a location purely to see how well the tag is doing, that's a difficult conversation to have. As well as that only, so yes, we've done it for range testing, but then it doesn't solve that issue of, let's say, a reference tag or a sentinel tag. Okay, this isn't to say you shouldn't do internal tagging at all. I'm not saying that at all, and in most cases, internal tagging is best. I think from a veterinary perspective, it's just a better option. It's better for that animal. You have less chance of tag loss, less chance of external injuries, etc. You just need to be aware that if you're using internal taggings, be careful with range testing, be careful with those reference or sentinel tags. Okay. All right, this gets into more assumptions of telemetry. We do telemetry to learn about these species and their biology and ecology. This means that we assume that those tagged animals act like untagged animals, that they're actually representative of the populations that we're tagging. How do you verify this? You know, you can't really track an untagged animal. That's the whole point of tagging the sucker. So one of the simplest places that we can start are simply holding studies. Right? And this is kind of the first step in this process. It's not perfect, but you can at least get a sense of, okay, if I take an animal and I put a tag in it or a dummy tag that has the same dimensions, and I take some animals that weren't tagged and I hold them in a tank or I hold them in an enclosure, I can see how well they do going through time. You can assess things such as tag loss. So that helps you from your tagged animals. How often do they just expel or lose the tag? You can assess mortality. If you measure and mark individuals, you can assess growth, but a little bit tougher. Note that I also have this what's called a sham surgery treatment. So a lot of these holding studies, it's not just tagged versus untagged. It's tagged, sham surgery, untagged. A sham surgery just simply means you're going through the entire process of a surgery, you just never put the tag in it. So that's the idea that, you know, it can be the presence of the tag impacts the fish. It could be that you cutting it open and doing surgery affects the fish, or it could be a combination of both. So you can do that. However, if you want to build on this, because right, this just gets at the idea, does it die or not? Or can it still eat? But right, that isn't the only things that are important to an animal, it's an environment. I study migratory fish. A migratory fish better be able to move. And so you can go on and do more intensive trials as well, and some of my predecessors at UBC have done this, where you not only do the surgery or the dummy tag and the sham surgery versus controls and look at tag loss, mortality, and growth, but also how well do they swim. Actually put them through experimental trials where you assess their burst swimming or their endurance swimming. And does it have an impact? However, I'll admit, I was trying to think about what would you do to a crab to try to understand better how it's influencing its ecology. So that's something I'm going to have to think about what the equivalent is. Maybe how well can it get into a hole? Because that seems to be what all of our crustaceans want to do, is go hide somewhere. Okay, now I'm going to get on a little bit of a rant, so I'm sorry. You only have 20 more minutes left of me, I swear. So tagging effects is often tied to this idea of tag burden, right? And so tag burden is simply what percentage of the animal's mass is attributed to that tag, or what percent of the tag is of the animal's mass. I don't know if I said that right, but I think you can follow. General in studies, we want to minimize this. In fact, we're often legally obligated to because of animal care. I mean, it is right in our IACUC standards that you must minimize and reduce harm. So part of that is reducing this tag burden on these animals, because hopefully our assumption that they act like untagged animals is valid. We also try to, in our studies, attempt to test or assess the impacts of tag burden on behavior. So this is after you get your data back. You can, for each animal, you can calculate what was its tag burden. So now each animal, among all the animals, you have a variability in tag burden. And then you're going to see, okay, well, did movement rate vary between the animals with the high tag burden and the low tag burden? Or if you're doing a mark recapture model and you're estimating survival, did those with a high tag burden have a lower survival than those with a low tag burden? Both of these aims individually are really good. They are things that we should do as researchers. However, when you put them together, it often prevents us from actually being able to do them both. And so hopefully I'll be able to explain this. Say you have a population of fish that you want to tag, and you are trying to minimize tag burden. Well, guess what? You're going to tag the biggest animals, right? We have to do this with juvenile salmon all the time. So we are high grading. We're taking out the big fat fish because those are what we want to tag so that we can minimize tag burden. And then we do these tests post hoc and we see, oh, well, there's no correlation between tag burden and travel speed or survival. But if you actually look at the variability in tag burden, it might only be 0.5% because the range is so narrow because we've high graded. So just because we don't see an effect of tag burden on the behavior of survival doesn't mean that it's not there. We might just not have pulled enough from a wide enough distribution to get statistical evidence for it. So we see this with our juvenile salmon, and one of our first indicators that tag burden might be influencing them was not through looking at survival, but actually through behavior. We not only used acoustic telemetry, but we used pit telemetry. And unfortunately, the pit array didn't work well enough for us to estimate survival, but we felt good enough to use these data for movement rates. And when we looked at the movement rates, we saw that age 1s, which are like half the size in terms of mass than the age 2s, had a similar migration rate between the age 1s with a pit tag versus the age 2s with an acoustic tag. Versus the age 2 fish with a smaller tag, a pit tag, moved more quickly. And so even if everything else about their behavior is the same and their fitness is the same, because survival is a time-based process, simply moving more slowly over a set migration distance is going to negatively impact migration survival. And so this is kind of the first clue to us that there is actually a tag burden effect because we could actually start to sample a wider distribution of tag burdens. This then led to us doing some research led by a former master's student, Christine Stevenson, where she sampled over a broader range using both the V4 and V7 tags and trying to quantify that tag burden effect. Okay. All right, so that's enough of my tag burden aside. Okay, so I've done a number of VPS studies. So I just wanted to touch on them a little bit, and we had a really good introduction to those early on. These are, again, a unique case, right? You need to have at least 3 or more receivers pick up a detection of an animal to get an estimated location back from it. So this requires your study geometry to have really close spacing, and I'm going to try to convince you to be really conservative with that. You need to have stable receiver locations. Right? A lot of our presence-absence work, it doesn't matter if that receiver moves 10, 15, 20, maybe more meters, right? Because as long as you can get in the general area where you have the acoustic release, it's going to pop up on the water, you'll see it type of thing. You can't have that with a VPS You need to make sure that these moorings stay as close to their original location as possible. And so you need to have really stable moorings, you need to have a lot of receivers in a close space, you have to pay VEMCO the processing fees, so you also just need a lot of money to do these studies just in general, or know people with money. I will also argue that you need to have really good questions to justify this cost, as well as the data. The idea of VPS data are really fascinating, right? I can see an animal move through time. That's really cool. I nerd out to it all the time. Actually assessing those statistically to try to answer a focused question is going to be more difficult. So again, you need to think about what your question is and does it require VPS data or not. And I love VPS data, but certainly they can't be used for everything. The first talk talked about the importance of being organized. I am going to reiterate it. You need to be especially organized for a VPS study. You need to know where every receiver and sync tag and reference tag is going to go out beforehand. I like to have my field teams, students, print out maps of everything with the stations named and kind of the serial numbers already in a waterproof notebook, that type of thing, so they can go out station to station to station. Have the GPS positions of where you want to put stuff already into your GPS or into the boat's GPS so that you can go station to station to station. You still need to log where they end up going, but you have that starting point. Again, ensure the moorings are stable to minimize movements. And so again, you get to laugh at me here a little bit. But don't laugh too hard, because this is Mo's graduate work, and she's in the room as well. And so for this project on Jonah crabs that we're doing a VPS, we were collaborating with some new collaborators for me, and they're crustacean ecologists. They're really good people, but they'd never done a VPS before. They'd always just done presence-absence work. And they've used these modified lobster traps for decades to do telemetry work. And they just said, yeah, they don't move, they're fine, type of thing. And I kept thinking, well, they just, we started throwing rocks in them, we started adding a bit more weight, because it just didn't seem hefty enough to deal with the tidal currents in the area. And what we learned is that, yes, these work really well for presence-absence, but they were not heavy enough for a VPS. And VEMCO did a fantastic job dealing with the over 50 movements of stations that we had over a 4-month period of this project. So a lesson learned is, again, these have to be really stable and really precise. They got to be really heavy, I think, in this system. You need to record everything. One that people tend to rush is the GPS. You get one receiver down, you just want to take a GPS reading and move on. Realize that there's generally a lag between the GPS and where you actually are. Just sit and chat for 30 seconds. Breathe, take a break, get prepared for the next site. Okay, now take your GPS measurement for that location. Take multiple ones if you can. Check on your receivers as often as possible. This will give you more GPS locations. Go back out to that buoy or location, take a new location, determine if the receivers have moved. Maybe the most important VPS tip is talk to VEMCO. They're really, really helpful. They understand how geometry of these triangles are going to affect your ability to get high-quality detections. They have some really nice checklists for VPS deployments that I share with my students and I've used personally before. Okay, we are really close, guys. We have 7 more slides. Do you think you can do it? We got 7 slides in 11 minutes. We're almost there. Okay, so this is just stepping way back. What do you need to do to be able to be successful in the field? Again, be organized. The first talk said it. Thank you. And I will reiterate it again and again and again. People think, based on their knowledge of me professionally and my work in telemetry, that I'm an organized human being. Because I go out to do a field telemetry study, and I know exactly what every receiver is, where it's going. All the data sheets are already set up. If we're doing biopsies of the animals to pair with the telemetry, it's all written up. All the vials are labeled, etc. That's because I know how important it is to telemetry. If you come and see my office, you would have a completely different opinion of me. Learn to love metadata. This is part of the organizational process. Work with OTN. Work with VEMCO. They will provide you tag info sheets. They will provide you info on what metadata standards are. And you can have some of this information even pre-filled potentially before you go out into the field. have gear lists. Have other people look over your gear lists. This depends upon the study. When I was working in Texas, they were day trips. We'd leave the university, we'd go out to the field site, we'd come back at 5. Maybe not as big of an issue, although I'll still give you an example. But let's say you're working up in British Columbia where you're going out to a field site for 4 weeks without coming back. You better have everything that you need, let alone going up to, let's say, the high Canadian Arctic. You better have everything that you need. So that process needs to start months in advance. You need to have other people with institutional knowledge look them over. Okay, I know this is hokey. I do a similar thing every Sunday morning. I just don't look as good at it when I'm playing old man basketball for my town rec center. But visualize what you're going to do before you do it. I had fellow students laughing at me, and deservedly so, but I would literally be doing a surgery at my desk of a fish. So as I'm making my gear list, so I can say, okay, I need the sutures, I need the needle holders, I need the anesthetic. And because literally, I'm just a visual person. And I'd get to that point where I'd reach for something and be like, crap, that's not on my gear list. And then I add it. And I would do that for every step of the research process, doing the receivers and the deployment, tagging animals, getting environmental conditions, etc. And so do this at several time periods to keep building your list. Okay, be organized as possible. I'm going to say it again and again and again until you're sick of me. Have backup plans. You need to constantly ask yourself, what if? Because it's really easy, because we're all critical thinkers, right? Most of us are in graduate school or working for academia or nonprofits or the government, industry. We're all really bright people, and so we have these really good detailed plans. But then sometimes we get out there and something doesn't work, and everything comes to a halt. So try to preempt that. Try to think about what happens if I get to this site that seems really important to me, and I can't get down that hill. Or, you know what, we see something in this location that means there's a lot of human activity here, and we think that there could be vandalism. We've run into those issues a lot. And so you need to try to try to preempt that as much as you can. This is kind of my obligatory slide on safety plans. Make sure that you are prepared for the environment you're about to go in. I think this is really important for kind of undergrads and graduate students, right? Because you've spent a lot of time in the classroom, you're really, really bright, and then you get this really exciting project. I'm going to go do telemetry in some remote area, and then depending upon what your institution does, what your advisor does, you might just be dumped into a situation, and you need to be really prepared for it. So thinking about what the hazards are, this is going to vary quite dramatically between, let's say, the rivers of British Columbia, where we require everyone to have swiftwater rescue training, much to my chagrin. That was the most terrifying 2 days of my life. What are going to be the potential for you to communicate with others if something goes wrong, both with your team, but also back at your home base, back at your institution? Where's the closest health care, and how do you get there? When we're in British Columbia, it was a several-hour drive or helicopter ride. For my folks in the Arctic, it's a lot more. And all of these safety considerations, if you're leading the field team, need to be communicated with them. They can't just show up and not know what to expect. That's on you to communicate with them. Okay, and I just want to end with a couple of slides so you can laugh at me more. So these are just some lessons learned in organization. So again, I did my master's at Texas A&M Galveston, and our field site, the image that I showed you that we're working on flounder and red drum, was Christmas Bay. It was only about an hour drive. It was an hour when you're towing a boat. And I can't tell you the number of times that, you want to get out nice and early onto the bay. We would meet on campus at 4 to 4:30 a.m. And then we would get our gear. We would drive down here, literally put the boat into the water, and then we'd realize we wouldn't have keys to the boat. Right? That's where that visualization, if I would have just visualized turning that darn key, I could have saved us a lot of time. Type of thing. There's nothing worse than having to drive back and go get it. So you think I would have learned that lesson during my master's. So go forward a few years to my PhD. Now we're driving a little bit further, right? We're talking about, a 10-hour drive, especially if I'm driving. And again, you get up nice and early because you want to get there. The last couple hours are on some pretty rough roads, so you don't love doing them in pitch black. And so you get up nice and early. I'm leading a field team of 2 trucks. We beat Vancouver traffic, because this is all just a nightmare for traffic. Don't miss it at all. And we get to a certain point, and I'm doing this visualization process way too late, and I realize I don't have any of these. Everyone in the room knows what these are and how bad of news that is. And unfortunately, we had already made it to Hope. You know, we had already gone a couple of hours. And this is where maybe another lesson is that, again, I'm not perfect, I'm not that smart, but I put a lot of effort into things and I try to treat people well. And that can get you some goodwill. So when I tell the 2 trucks, guys, this is what I did, I'm sorry, I screwed up, they grin and bear it, and they say, that's okay, Nathan, we'll take this 1 truck up, you go back and get it. I then also call one of my lab mates who's back at the University of British Columbia, a really eager guy by the name of Nick Burnett, who's now with BC Hydro, I believe, and I say, Nick, I know you're in the office early to get work done, but can you drive out 2 of these magnet keys and meet us? And he said, yep, that's fine, you've helped me plenty of times, Nathan. And we met almost in the middle, a little beyond Abbotsford, and we were only a couple hours late instead of maybe 4 or 5 hours late. One last message. In the field, you're going to be pulling long days. You know, when we were doing our Chilco Lake work, we were often pulling very long days, not finishing till well after dark, and I would still make sure that I backed my data up each time. Now, granted, I'm a bit old school, so this was literal backing up of data and typing it in the notebooks, and then working with some of the younger crowd, they literally just pull out their smartphone and take a photo of every page. It's like, well, that was way easier. Okay, so I have 2 minutes left. I think the timing's pretty good, so here are just some conclusions for you. There is no one-size-fits-all strategy to telemetry. Right? The geometry, the programming, the tag type, how you deploy these things, the length of the study are all up to you. And this is really exciting because you can make it or optimize it to what you need, but it's also certainly a little daunting. Focus your decisions on questions. And what data do you need to answer those questions, and how likely are you able to actually get those data? I push you guys to improve upon my mistakes and to keep thinking about new ways that we can develop these arrays and deploy our studies and be successful. Remember that no study is perfect. You're just trying, it's an optimization problem. So don't think, well, crap, I can't do the proper Sentinel tags, or you know what, the Sentinel tags are going to have to be external. That's okay. You just need to acknowledge there's a weakness there and try to think about how that's going to influence your data. It's just like any other study, right? You always have that section in your discussion that's talking about all of the weaknesses of your work and how that might or might not impact your results and interpretations. Telemetry is the same. Rely on others who've done this, both at your institute and maybe the folks in this room. And I'd like to think I'm not a terrible scientist, but I certainly do a lot of silly things. So realize you're going to do silly things too, and that's all right. You're here to learn. Most of you are graduate students. You're a student. You're supposed to learn. You're supposed to screw up, okay? I'm not. I'm a professor now. So thank you. I'm sorry that things ran. I would have liked to have built in a bit more time for questions, but I am around all day today and tomorrow, so please feel free to reach out to me. There's my Twitter handle and email as well. So thank you guys so much for letting me be here.